Today, we will be explaining how DNA replication and protein synthesis work. To start us off, we'll begin by discussing DNA structure. A nucleotide is made up of three parts: phosphate, pentose sugar, and a nitrogenous base, either adenine, thymine, cytosine, or guanine, or uracil if we're talking about RNA. The 5 carbon atoms within the pentose sugar have to be in specific spots. The first carbon is the one closest to the base. They then go clockwise from there. To explain which way a strand is going, we either say it's going 3' to 5' or 5' to 3'. DNA is always read by enzymes in the 3 to 5' direction, in both DNA replication and protein synthesis. On the same note, DNA strands are said to be anti-parallel, meaning they go different ways. Let's start with DNA replication. This is needed for mitosis. It occurs within the nucleus. When replication is complete, a new copy of DNA is paired with an original copy of DNA. This means the replication of DNA is semi-conservative. First of all, DNA helicase breaks through the hydrogen bonds between complementary bases. It essentially unzips the two strands. Next, DNA gyrase stabilizes the DNA molecule by reducing the torsional strain caused by helicase. It essentially flattens out the DNA molecule. The single-stranded binding proteins, or SSBs, prevent the separated strands from reannealing. Primase generates a short RNA sequence, which is called a primer, on each strand to start DNA synthesis. This is really important because if there were no primers, then DNA polymerase III wouldn't know where to start. Polymerase can only build strands in the 5' carbon to 3' carbon direction. Since the strands are anti-parallel, this causes the creation of a leading strand and a lagging strand. As the DNA unwinds, since DNA polymerase III can only build in one direction, the lagging strand has to try to keep up, and it skips back and forth. Meanwhile, the leading strand is continuously synthesized by DNA polymerase. This causes sections called Okazaki fragments. To fill in these gaps between the fragments, DNA ligase creates a phosphodiester bond between the 3' sugar and the phosphate group. DNA polymerase I then needs to go back over and replace all the RNA primers with DNA nucleotides. At the end of it all, DNA polymerase can also proofread the replication of DNA, in a sense, removing nucleotides from the end of a strand when a mismatched base is discovered, but this is extremely rare.
Next, we are going to be talking about protein synthesis. Protein synthesis has 2 major steps involved: transcription and then translation. Transcription happens inside of the nucleus. What transcription is trying to achieve is using a gene's DNA sequence to make a copied RNA version of it. The RNA copy or transcript carries the information required to create a polypeptide for a protein coding gene. That information is stored in the mRNA, in which the M stands for message. The sense strand is a DNA strand with the same base sequence as the mRNA. RNA does not copy it. The antisense strand is a DNA strand that does not have the same base sequence as the mRNA. RNA does copy it. Here is the process for forming this mRNA. RNA polymerase, which is an enzyme, is a crucial factor for this process. RNA polymerase binds to a site on DNA at the start of a gene. RNA polymerase goes along the gene, separating DNA into single strands and pairing up RNA nucleotides with the complementary bases. It's uracil with thymine instead of adenine. RNA polymerase forms covalent bonds between the RNA nucleotides. The RNA separates from the DNA as the double helix reforms. The process for transcription is done at the end of the gene. Now mRNA is formed. Once the mRNA is complete and ready to leave the nucleus through nuclear pores, it enters the cytoplasm and attaches to the small subunit of ribosome. Now translation comes into play.
Next up is translation. Translation is a process where protein is synthesized from the information contained in a molecule of messenger RNA. The information is carried to ribosomes in order to build the correct polypeptide. Translation occurs in ribosomes. Each ribosome has a small and large subunit and is a complex molecule composed of rRNA. Let's look into tRNA. tRNA molecules are molecules in the cytoplasm that carry an amino acid each. Don't forget that amino acids are the building blocks. tRNA has a 3-base anticodon that binds to its complementary codon on mRNA. The sequences found on mRNA are read by using genetic codes. Genetic codes are a combination of 3-letter combinations of nitrogenous bases called codons, using A, U, C, or G, each of which corresponds with a specific amino acid. The first codon to bind to mRNA is the start codon, AUG. It signals the start of protein synthesis. It corresponds to the amino acid methionine. tRNA attaches to the mRNA, bringing the amino acid methionine to the ribosome to start the polypeptide chain. While still being attached, a second tRNA molecule comes into play and attaches to the mRNA at a second bonding site, hauling the amino acid that corresponds to the next mRNA codon. The bonding of two amino acids occur by condensation reaction, forming a covalent peptide bond and making a new dipeptide. Then, the first bond between the amino acid and its tRNA is broken down by an enzyme. Ribosomes slide along the mRNA, moving down one codon by releasing the used-up tRNA back into the cytoplasm. This process continues until one of the 3 stop codons, UAA, UGA, and UAG, is reached. At the end, the ribosome moves along the mRNA. The polypeptide will fall off and be released into cytoplasm. This is now a protein. It gets edited and or folded, then it performs its function in the body.